for Food Safety and Technology, and is now the Institute for Food Safety and Health. But today she's an adjunct professor in the Department of Food Science and Nutrition, where we welcome her expertise and years of experience. It's an opportunity for the students to learn things even beyond what you can find in textbooks. Cindy has done a very nice job in circulating her bio, so I won't continue much longer. Time is now suddenly getting short, and she has lots to communicate with us today. So Dr. Tatiana Koutchma, welcome, and the floor is yours.
Thank you very much, Britt. Thank you very much, Cindy, for organizing this webinar. And I'm excited to share, whatever knowledge and experience I have in light technologies. And also I'm hopeful that this information will be very useful for students. And I know because many students are taking advanced food processing course, and I hope that they will learn about the recent developments in light-based technologies for food, beverages, and grain processing. And the title of my presentation is Capabilities of Ultraviolet Light Technologies for Food, Beverages, and Grain Applications. As Britt said, I'm right now with Agriculture and Agri-Food Canada, which is one of the 4 regulatory agencies in Canada that regulate food, actually. So just for information, in US, there are 2 agencies, FDA and USDA. In Canada, there are 4 agencies. This is Health Canada, Public Health Agency of Canada, then CFIA, so it's Canadian Food Inspection Agency, and Agriculture and Agri-Food Canada. All these 4 agencies having different functions to control, to regulate, and to inspect, different food facilities. And then Agriculture and Agri-Food Canada, we are mainly dealing with agri-food and, and with research and development and dealing with commercialization of these technologies. So we are one Global Food Research Center, which is located not far from Toronto, is one of 17 such centers. And there are 3 centers that are dealing strictly with the food. So this is a picture, and this is a location, of this facility. So it's about 100 kilometers. I'm in kilometers, when I'm talking about Canada. So it's 100 kilometers from Toronto. So, and also in Guelph, there is a University of Guelph. So in Guelph, there are a lot of government organization, and we are working with other government partners, with the university and industry, and to improve food safety, sustainability. Right now, sustainability is a very important issue, quality and nutrition of the food. So in when I joined Canada, this Agriculture and Agri-Food Canada, actually, I started a program on novel food process technologies, and UV technologies was one of the technology of the interest at this time. So actually, so since that time, it's already 15 years, so I'm working at this technology and high pressure technology. And basically, also the advantage of our facility that we have a BSL-2 pilot plant where we can validate units, pilot scale units, and using pathogenic organisms. So our program is actually based on, so in our program, we are looking at efficiency of different UV light sources, such as low pressure, medium pressure lamps, eczema lamps, now pulse lamp, and light emitting diodes, LEDs. You will see it on my slides, LEDs. And we are using these technologies for preservation and improving safety of drinks and beverages. Like I said already, validation of continuous UV systems for low UVT liquid and foods and beverages. And we are looking at different effects of light technologies on nutritional properties of different foods and beverages, including cold pressed juices. We just completed this work. So also we use UV light to improve toxicological safety of apple or grain products. And we looked, because the chemical safety, one of the very important issues of a chemical safety. So we are looking at UV light effects on generation of chemical compounds in foods like HMF. And recently we started working with UV LEDs for treatment of fresh produce. And on the left here, you can see some of the equipment we have. And recently we've got a UV conveyor unit when we have a possibility to change the different UV sources. So, okay, so in my presentation, I have 45 minutes, and I know there is some of you familiar with the technology, some of you are less familiar. So I will start with very briefly with reviewing of fundamental principles, advantage, and regulatory status of UV light technologies and its safety. Also, I would like to provide you an update of the current state of the development of monochromatic and polychromatic UVC light sources that are conventionally used in industry and they're emerging. And also, I would like to introduce you to some food applications of UV. And this includes preservation of cold-pressed fruit and vegetable juices, post-harvest treatment of grains, and especially corn to reduce fungal and toxicological contamination, and also some results of how LEDs can be used for surface treatment of fresh produce. So this is basically the first introduction in light technology. Actually, they're based on electromagnetic waves, and it's the light, and it's part of electromagnetic spectrum between X-rays and infrared light, and they cover actually the range between 100 and almost 1,000 nanometers. And light technologies, they include ultraviolet light, visible light, and that's why I included here blue light, and infrared. Sometimes infrared technologies are also referred as a light technology. So because of the different location of this technology at the UV and the spectrum, the light photons, they have different energy and different wavelengths. And in this drawing, you can see that there are different sections of UV light spectrum, UVA, UVB, and UVC. And actually, they have different effects on the organism. So blue light between 400 and 450 nanometers, it causes photodynamic inactivation. UVA between 315 to 400, they cause oxidative damage to DNA, partly proteins and lipids through formation of reactive oxidative species. And UVB, it's located between 280 to 315 nanometers. It also can damage DNA and activates various repair enzymes via ROS formation. And definitely UVC, that is between 180 and 280 nanometers. UVC has highest germicidal efficacy and mainly damages DNA via pyrimidine dimerization. So this is very brief mechanism of inactivation of these three sections of UVC, of UV light. So, and why actually, UV light is, why there is such an interest in light technologies, and why it can be used more and more in industry. And the main reason, because UV light is novel, non-invasive food preservation technology, and the industry right now is looking at to find non-thermal, non-ionizing, non-chemical, organic technologies that has relatively low initial capital investment, and they require smaller space and lower operating costs. They can be energy efficient and continuous. So, and these are all the requirements that actually light technology can meet, because it's physical. And what is very important, it's non-ionizing, so the energy of photon doesn't cause the damage of chemical bonds. And what is also important, UV light technology, they're effective against different microbial hazards, and this include water and airborne pathogens, and also food and food-borne pathogen, and also spoilage organisms. So these are key milestone in the development of the light technologies. So if you look at the, at this milestone that I listed here, light, UV light was discovered in 1801 as a light beyond violet, and only almost 100 years later, the bactericidal action of light was found. And in order to build a lamp, silica quartz tube was developed in 1906. And then when the first tube was developed, quartz tube, because it's transparent for UV light, and usual glass, it's opaque for the light rays. So since 1930, UV light technology was well developed for water and air disinfection. So, and it has UV light technology has quite a rich history for water and air treatment. However, for food and beverage treatment, it's quite a new technology, and it was approved only in 2001 by US FDA for applications for juices, and then it's also as an application followed by approval of Health Canada for juice applications. And the very important milestone was introduction of LEDs for water treatments and instead of lamps. And in 2016, European Food Safety Agency approved UV light for milk treatment. And then in 2018, Health Canada also approved some other applications for liquids such as blood treatment. And in 2022, there is actually, there was a boom, in the development of different new novel UV sources. And this included sources 222 nanometers, pulse light, and so on. So, and of course, in order to use this light-based technology on, in the food enterprises, of course, this technology has to be approved by regulatory agencies. And this is a very brief slide that says about international regulatory status of UV. And on the left, you can see that in USA, FDA treats UV light as a food additives. And in Canada, European Union, New Zealand, and Australia, the foods that are treated using light technologies are considered novel foods. So, and this is a list of some products that are currently approved for, that can be used, that can be treated using UV light. So, like I said, it's juice, milk, mushrooms, can be treated with pulse light to generate vitamin D, bread and yeast, some surface treatments, and of course, water. So, okay, and this slide actually shows the very fast growth of applications of UV light technology in the last 2 years during pandemics, because it was found effective, the light technologies, they were found effective against COVID. And there was a lot of research going in the last 2 or 3 years, when a lot of experimentation. So, and this slide actually shows the we see and virus actually, emerged as a new area of research with a with a very big growth of different publications in this area. So let's talk about UV light technologies. So light-based technology actually, they can be continuous and they can be pulsed UV, they can be pulsed broadband lights, and also there are advanced photooxidation technology. So in continuous UV light, there are monochromatic lamps that can emit light at 253 or 222 or 282 nanometers, that is almost on a border with UVB. Then there are broadband polychromatic lamps that emit light between 260 100 nanometers, and they have multiple peaks here. You can see on this plot below. So low pressure lamps, they're using mercury as a filler for the tube, and it has only one peak at 254 nanometers, and it can also emit some light at 185 nanometers that can create some ozone. And pulse light broadband lamps, xenon, mainly xenon lamps, they emit light between 200 up to 1,000 nanometers, including also infrared part of the spectrum. So yeah, and one more important source of continuous light. So I mentioned already LEDs. They can include UVA, UVB, and UVC, and they're very, very tiny, very small. You can see the picture here compared to the conventional lamp. And the advantage of LED that can emit light at selected wavelengths. So, and these are examples of emission spectrum of low pressure lamps at 184, like I mentioned, 254 nanometers here. And this is the wavelengths that actually approved for use for food by US, FDA, and Health Canada and European Agency. Then we have medium pressure mercury lamp here, between that emits light between 200 and 600 nanometers. And it's mainly used for water disinfection, but approved for sterilization of rinsing water in dairy operations. And like I said, UV LEDs. And recently there is a huge interest in eczema lamps that emit light at 222 nanometers because of the safe applications of this lamp for humans. And recently we started working with pulse electronic UVC lamps that emit lights that can emit light strictly in UVC region. So yeah, so like I said, it's very important to consider that UVC light, the safety of UVC lights for humans, and potentially UVC radiation can affect skin and vision. So, and that's why UVC equipment, this is category C equipment, and UVC radiation can cause some skin cancer, and also can damage eyes, vision, and cause cataract. So, and because of this, it's very important to know that protection equipment should be used all the time when working with UV light sources. And this include, of course, gloves, thin and flexible gloves, and also the face shield. So, and this is important because very often, we are not talking about the safety of the technology. So let's talk about applications of UV light for foods. So like I said, for food applications, UVC light is quite novel technologies and has a variety of applications. And I grouped them in main 4 categories: for safety, for preservation, and shelf life extension, to improve functionality of foods. And also it can be used to destruct chemicals. So for safety applications, mainly on food plants. So UVC light can be used to improve quality of air, surfaces, and water. So because there are a variety of different sources of contamination on food plants that include animals, people, some equipment, pests, and so on. And so UV light is very effective means to use against airborne, waterborne, foodborne, and spoilage organisms at food plants. So, and there are a lot of different known applications of UVC light for air and water treatments. For non-food contact surfaces, that includes walls, ceilings, and floors. After, typically, it's this treatment of UVC light occurs after some cleaning or washing of this non-food contact surfaces. And also UVC light can be used for food contact surfaces. That includes conveyor belts, packaging, and the equipment surfaces. So, and also it can be used to improve the microbiological quality of different foods, pre-packaging or in post-packaging operations. And here on this drawing, you can see how, the raw food, can be exposed to the microorganism and can be contaminated, or, can get some contamination through the air. So, and like I said, there is like a lot of new applications right now, and microbial resistance to UV is a very important factors for effective use of UV light. And actually, like I said, in plants and food plants, the contamination of air, water, and food can be reduced using UV light. And actually, it's important to remember that the resistance of different microorganism groups varies in this different medium. So the spores are considered the most, mold spores are considered the most resistant in the air. And this resistance depends on relative humidity or temperature of the air. In the water, adenoviruses, they are considered the most resistant organism. And cryptosporidium parasites, they are the least resistant. And turbidity of water, plays a very important role in activation of these organisms. So it's critical parameters. In foods, so far the mold spores, they are the most resistant organisms, and of course the composition of food, the pH, water activity, and other parameters will affect this resistance. So these are the examples of different UV units for treatment of air that can be used in food facilities, and these are, they can be mounted on the ceiling. And this application is such applications are safe for people, so they continue working in this facility. They can be mounted on the wall, or they can be mounted in the duct system. Or they can be used to disinfect containers and to make it like I said, to reduce the cross contamination in food facility. So the next applications of UV light is for liquids and for beverages. And they can be used for preservation of liquid and beverages. And the ideal application would be to use it for pasteurization of raw milk, juices, or other soft beverages. But more often right now, the light technologies that are used as adjunct to pasteurization of these products. For example, ultra-high treatment of milk. This is actually application was approved by European Union. Also, it can be used to improve microbiological quality of cheese milk as a non-thermal option, and teas, and for increasing shelf life of juices, different protein ingredients, and syrups, especially sugar syrups. And what is the difference between applications UV light for water treatment and for beverages treatment, for food treatment? It's of course the UV transmittance of different food fluids. And if UVT of water is almost 99%, so compared to water, the UVT of juice is about only 1%. So it means that the UV light can be absorbed in very, very, thin layer. And for water treatment, actually, UV light, 99% of the light can go through the sample, depending on the thickness. So, and Beer law, it's actually describes, it's a formula how to determine the distribution of UV light depending on the transmittance or absorbance of the liquid. So, and because of this higher absorption properties of liquids, the evaluation of UV dose is quite complex for food applications, because we have different types of energy. And this actually drawing tries to illustrate this, because we have power supplies and lamp power that applies UVC energy for treatment, and we have incident fluence on the surface, and then this incident fluence can be absorbed by the liquid and or scattered by the liquid, or can be absorbed and can be delivered to the microorganism. So in case of water treatment, the UV mainly transmitted, like 90% of energy can be transmitted. That is opposite to the beverages and other food liquids. So maximum 30% or much less, can be transmitted. So it's very important to in order to determine the energy requirements and dose requirements, to determine the UV dose for different applications. And in the water treatment, there is a standardized method to using animated beam tube, where the water is actually, mixed in a Petri dish, and we can measure the incident fluence and evaluate the absorbed fluence, and depending on the fluence, we are building survival curves, and this allows us to determine the effective dose to reduce to get one log reduction. So, and knowing the dose for one log reduction and knowing the total or required log reduction, we can determine the required UV dose. So basically, this is the main formula for pilot scale multiple lamp unit for determination of the dose. So decimal reduction dose is typically multiplied by required log reduction. So, and this gives us the effective UV dose to be delivered in the process, and the process, can be different. So, knowing this, we need to also to determine different critical parameters, product and process parameters that can impact the delivery of this dose. So, among product parameters, pH, water activity, and composition of the product is very important, because some ingredients actually they can absorb the UV light. Also, the viscosity of food liquids it's much higher than water, and like I said already, UVT plays a very important role. So, and also process parameters such as the flow, laminar or turbulent, light source intensity, and UV dose itself that can be applied, incident, absorbed, and delivered. These are the main critical parameters for the treatment of liquids and beverages. And these are examples of a turbulent flow UV system, and turbulent flow is mainly used in industry in order to deliver the liquid to the source, because like I said, the penetration depth is very, very small. So the very good mixing is a must, and these are different designs of commercial systems. And typically they're using coil-based reactors to mix static systems to mix the liquid, or very thin film layer, or dynamic mixing is used in order to achieve the turbulent flow. So, and when we are establishing an operational dose for such commercial system, we need also, in addition to reduction of the most resistant pathogenic organisms, we need to look at shelf life of the product, effects of UV light on enzymes, like I said, shelf life achieved, effects of this dose on quality parameters and nutrients, and also effects on the sensory and appearance. And this kind of optimization of the dose will give us the optimum operational UV dose at 100% of output that can be used in commercial operations. However, there are very limited studies that give us that can consider all these factors. And like I said, one of the advantage of UV light is high quality of products. So we need to look also at color, physical property, vitamins, enzyme, and so on. And typically, we did some review of these effects in reported studies. So typically, there is no effect of UV light on color, or there can be some beneficial effect. For example, on chlorophyll particles, they become brighter. No effect on physical properties. And vitamins, degradation vitamins much less, and after heat treatment about 22 maximum 30% of degradation, and very limited effects on polyphenol degradations and antioxidants degradation. So, and of course, like I said, UV light is there is very limited use for pasteurization. I don't know much about this, about this applications, because of the different issues of microbiological or chemical nature. And we need to know the most UV resistant pathogen organism. We need to know the UV dose to achieve the required log reduction. We need to select the correct surrogate organism. We need to conduct validation study, verification study, and also to look at some photoreactivation effects. So, and also because this is novel treatment and it can cause some chemical changes in the food or beverage. So the chemical effects of such treatments are also very important. So we looked at effects on the composition, oxidation of fatty acids, protein oxidations, formation of furans, like effects on nutrients, formation of volatile compounds, formation of flavors, toxicological concerns, and allergens. So, so there is still a lot of knowledge that is required, in order to fully use the capability of this technology for beverages and juice treatment. So in our research, we also looked at applications of UV to reduce fungal toxins in grains. And the reason for this is that grains are, one of the, very important production product, and that brings a huge revenue to Canada. And however, 20% of this world grain is contaminated with mycotoxins, and there are huge revenue loss. And two fungal strains that are largely responsible for these losses: Fusarium, Graminearium, and Penicillium. And you can see that, the appearance of, of this grain after, they were, when they got contaminated by these organisms. So there are different mycotoxins that are potentially cancerogenic that are produced by these fungi. And this is DON, zearalenone, ZEN, and ochratoxins. And what is interesting, that they have an absorption spectrum somewhere in the middle of UV spectrum. That's why we decided to use UV to reduce the level of this organisms that produce this mycotoxins, and also to look at the dose that reduce the content of this mycotoxins. So, and what we found that actually, this molds, they're very sensitive to UV treatment, but the toxins, required much higher dose, 100 millijoules versus like 10,000 millijoules, because this is chemical. We are looking at different locations and the production of gray where this technology can be used to reduce this contamination. So in order to treat corn, we have a unit that allows us to treat this corn from both sides. And here you can see the reduction of the fusarium on the surfaces of corn. And we found that actually, the concentration of fusarium, can be reduced significantly, by using UV light. So, and right now we are working at the process that allow us, to treat corn in continuous mode. So, and we will be using, different alternative UV light sources, including medium pressure lamp and pulse light, in order to be able to deliver the high dose and, in a very short period of time. So, and I have about 5 minutes, 10 minutes left. So I will be briefly talking about UV LEDs for surface treatment. And the reason for this is because this is a very novel source of UV light, and this is solid state semiconductor lighting devices. And they can emit light in very narrow emission wavelengths in UVC, UVB, and UVA range. So they are very small and robust and can be tuned. And actually, the LEDs and UVA and UVB range, they have a very long lifetime by up to 100,000 and high efficiency. But UVC LEDs, they didn't reach this efficiency yet, and and that's why we are comparing the technical parameters of UVC LEDs with UVA, UVB, and low pressure lamps. And right now, of course, the efficiency of UVC LED is quite, it's lower than lamps, 30% versus 10%. However, there is a huge potential to increase this efficiency because of, very fast development in this area. And this time, the power output and efficiency is rapidly increasing, and the cost of LEDs is decreasing. So this is very important for LED production. And actually, LED chips, this is the heart of these technologies, and they are used, them made from wafers. And, like I said, these are semiconductors, and you can read more about this. It's a lot of information of this. In order to use this tiny LEDs, they have to be packaged, and there are different packed arrays in order to achieve the high efficiency. And these are examples of, different mount of the LEDs on the boards. And actually, the efficacy of LED chips and packages depends on a lot of parameters, and thermal management, mechanical mounting, and controls, and some additional optics. But when all these factors are considered, so these chips can be used in LED systems. So from the chip to the package to LED systems, and you can see the size of this LED reactor for water treatment, and they can be used for conveyors, and this application actually for treatment of fruits and vegetables, and for air treatment. So, and I know that many familiar with LEDs already, and there are a lot of commercial applications and lighting and horticulture and machine vision and curing. And now it's time for food and beverage industry to use this UV sources. And this is very interesting applications actually in Samsung refrigerator. Then it can be used for water purification, this technology, for ice disinfection in washing machines, and for odor removing and for disinfection of odor. And there is a lot of interest from manufacturers of refrigerator to use this technology to increase the shelf life of the products. So, and another advantages of LEDs that they can operate in a cold environment, and that can be very effective sanitation measure in food chain, and it can extend storability of fresh produce. And this is a table that I made to show where visible LEDs and 400, 760 nanometers can be used, and UV LEDs. So basically, visible LEDs are already in practical applications, and UVB, UVC, and UVA LEDs, they can be used for different purposes to enhance nutritional content, for disinfection, and for spoilage control. So we have a lot of different LED resources in our pilot plant, and we started our research with using collimated beam unit for LEDs with multiple wavelengths shown here. Then we have a chamber for the large samples for the whole fruit or vegetable treatment. And we have thin film continuous units for liquid treatments, also using different multiple wavelengths, 265, 275, 285. And the task is to find the most effective wavelengths for food pathogens. And from this point of view, we are looking at engineering aspects of the technology and microbiological aspects. So, and by applications, of course, the efficacy defined by the wavelengths, the UV dose required, the pathogen of concern, and UV irradiance and treatment times, because the irradiance can differ very much for different wavelengths. And also by the LED technology itself. So by the cost, the power output, and efficacy of LED packages, optical geometry, and of course, uniformity of treatment, or sometimes combination of wavelengths can be used very effectively. So also for biological consideration, we need to find the most effective wavelengths for different organisms. And this is called action spectra. So it shows the spectral effectiveness of photobiological or photochemical process of microbial inactivation. And actually, this is for waterborne pathogen. And we built a range of action spectrum for foodborne pathogens, for Listeria, for E. coli, for Salmonella. And what we found that 254 nanometers that is used in low pressure lamps, it's not the most germicidal wavelengths. And sometimes 265 is, more effective for example, for Listeria, but 259 somewhere, it's more effective for Salmonella or for E. coli, for pathogenic strains. So this is very important to understand this dependency. And because of this, sometimes they combine application of UV LEDs. They have a lot of sense when UVA, UVB, and UVC LEDs can be combined to suppress photorepairabilities of cells, and they can result of synergistic effects. So in advanced oxidation process, UV LEDs can be very successfully combined with photocatalysis, with ozone, hydrogen peroxide, and chloride. So that's called advanced oxidation process, and there is a lot of interest in such a process right now. So yeah, so taking into account all these considerations, engineering, cost, and microbiological. So looking at the UV LED market, right now right now we are thinking that 275 to 77 nanometers is the most efficient LEDs, and we use these LEDs for reducing penicillium expansum contamination on apple's skin. And you can see the log reduction that we achieved when we treated apples in this LED box. So we achieved approximately 3 log reduction at 277. That is comparable to chlorine treatment. And potentially, the conclusion was that UV treatment at 277 has potential to mitigate spoilage due to blue mold on apples when it treated with prior any mechanical damage. So you can see the pictures of apples treated, no treated apples, control, treated with low pressure lamps, and this treated with LEDs at 277 nanometers. So they look, much better than control systems. And also, we use it for treatment of fresh produce, and we achieved 2.6 log reduction on lettuce of E. coli, and Listeria on apples. We also achieved almost about 2 log reduction when we used LEDs, and the dose up to 500 millijoules. So, and this is our current project when we are using LED for treatment beverages to 3 wavelengths. And we are, we have such a miniature installation, and we show that this 255 and 285 nanometers wavelengths can be used for treatment of coconut water and blue lemonade very effectively. So, and as a result of our research, we found that UV LED are capable of an activation of pathogenic bacteria and spoilage organisms in model system on fresh produce. And the ability to emit light at different wavelengths lead to generated synergistic effect between multiple wavelengths. And specifically, we found the synergistic effect between 259 and 289 nanometers. And of course, cost, power output, and activation efficacy of UVC LED is very important consideration when selecting UV treatment wavelengths. So, and these are different resources that were published in 2019 and 2022 that actually will provide much more information about these technologies. And yeah, so it's ultraviolet LEDs for photo applications and validation of food preservation using novel technology. And this there is a chapter on UV technology as well. So thank you very much. I think I have 5 minutes over my time, and this is my contact information. So thank you very much for the interest. So, and this is my last slide.
All right. Well, thank you very much. We do have about 5 minutes for Q&A.
Yep.
Are there any questions from the audience for Dr. Koutchma? Well, maybe while, go ahead. Who is that?
Hi, Catherine. This is Catherine. So I saw that you showed in your chart that the visible light options, or the LED light options, I think, that could even be used for like hydroponics or during growth cycle. And that was the question I had, which was, which are there any, or how do these work to sort of interrupt the growth of pathological organisms without damaging like the healthy tissue of plants while they're growing? And are there options, or are people looking into using them like at the site of harvest?
Let me clarify the question. Are you talking about UV light or blue light?
I'm not sure. You had a slide where you had the 2 different types. I want to say, I thought it maybe said LED light. It was like a chart, and there were 2 different types that kind of cascaded down. Let's see.
What do you remember? Because
you said you had put together the chart. You were like, I made this chart. Now I can't remember.
Okay.
But I guess any type of light. The question I have is like, how can they be used for hydroponics and on plants that are still, or products that essentially are still living?
Yeah. So because I think the answer here will be, okay, when you're using for plant pathogen, or when you're using for the hydroponics to treat this liquids that with the nutrients. So it will be 2 different applications, right? And a different organisms of concern. And of course there will be different wavelengths, and I believe there will be different efficacy of LEDs. And I know for the plants, B and UV LEDs A and B, they're mostly used. But for an activation or reduction, of microorganisms, mainly UVC LEDs, are used. And typically, like I said, there is very limited effect on the nutrients itself. So, and that's why it's used. And typically, this nutrient solutions are quite transparent, I believe, so that's why they can be used. I don't know if I'm answering your question, if I understood the question correctly. So, yeah.
Yeah, I think you did. It makes sense to me that for hydroponics, it would be something you're using to treat like the water and the nutrients with, as
Yes.
opposed to like directly applying it to the.
Yes, to the plant, you know. So, and I think, you know, like I showed, I think, you know, the optimization of the dose, you know, for all application, that's very important, you know. So the idea is like, you know, okay, to have a maximal effect on the microorganism and the minimum effect on the nutrient itself. And this is where the choice of, you know, different wavelengths, the right choice of the different wavelengths is very important, you know. And we are actually doing this right now, you know, like, you know, we are debating what is like better, 281, you know, or 224, and like, you know, 254, because 254 is very close to the, for example, to vitamin absorption spectrum, and 281 is a little bit further, so it won't absorb it and it won't destroy it. So all this consideration should be considered, you know, so should be taken into account. Yeah.
Okay, we might have time for one more question, quick question.
Yeah. Yeah.
All right, Jason is just saying, thank you very much.
Okay.
I don't know if there is,
So this is TJ Fu. Hi,
TJ,
Tatiana.
thank you, TJ.
Yeah, hi, Tatiana. It's good to see you and do some wonderful work. I'm curious about this strawberry. So I know UV is a surface technology, and then strawberry, you have all those valley and hill in the surface. So I don't know what's the penetration range that you can actually effectively penetrate enough depth so that everything on the surface will be killed. So is there any
yeah, I think,
thought?
you know, there is no no penetration, you know, at all. You know, this is just like, you know, strictly surface treatment. And again, you know, it depends very much on wavelengths that, you know, can be effective against, you know, the specific organisms like on the surface. You know, for fungi, you know, we found like, you know, 277, you know, can be sufficient enough, you know, without penetrating at all.
Yeah, but bacteria can hide inside the surface of
Yes.
strawberry or even lettuce.
Yes.
So how do you
Yes.
solve those problems?
Very often, you know, they're like, you know, like I mentioned, advanced food oxidation technologies, you know, so they're using ozone or some chemicals, you know, in a very small concentration if there is a need, you know, or like, you know, sometimes the light source that can generate, you know, some ozone also can be used, you know, for this purpose if you know, want to enhance the effect. But typically what I saw, like, you know, I think, you know, depending on wavelengths, you know, up to 2, in the best case scenario, 3 log reduction can be, you know, obtained, you know, with LEDs for this.
Okay, thanks.
Yeah.
All right, well, we're going to wrap up this session today. Thank you again, Dr. Koutchma, and thank you all for attending. I know a couple students need to take off for class, and others probably have other commitments. So thank you again, and we'll see you next week.
Okay. Okay. Thank you. Thank you very much.
So much.
Thank you.
Thank you.
Thanks again, Tatiana. That was fabulous. Really
Thank you.
appreciate your time. Really good. All right, we'll see you soon.